Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from Department of Biochemistry. Today we are going to start with the first lecture of regulation of gene expression. Gene expression is the production of gene product, functional gene product from the DNA. These gene products, it may be either in the form of RNA or in the form of protein. The first step in the gene expression is the production of RNA, that is the transcription. These genes, whether they are transcribed and they are translated, they are regulated, but not all the genes they are regulated. The genes which are described as constitutive, they encode the product required for the basic cellular functions, and so are continually expressed. They are the housekeeping genes. They are produced, they are expressed at constant level. All the time they are produced at constant level. But the regulated genes, they are expressed only under certain conditions and only in certain subset of cells. For example, it may be in some organs, for example hepatocytes. This gives the cell to have the control over its structure and function, and it is also the basis for the cell differentiation, morphogenesis and adaptability of any organism. Regulatory sequences and molecules. What are regulatory sequences and molecules? First of all, we will see that how the prokaryotic genes they are regulated and how the eukaryotic genes they are regulated. See the upper diagram where the prokaryotes is written. For the most genes, the main site of control is the transcription of the DNA to RNA. So in the prokaryotes, primarily the regulated genes or the regulation is at the transcription level. Also in the eukaryote, transcription has the main sites of regulation, but in the eukaryotes, the gene expression also controlled at post-transcriptional and post-translational process. Now, what are the molecules or what are the sequences which control the regulation? Regulation of transcription, the initial step in all the gene expression, is controlled by regulatory sequences. They are present within the DNA. It may be in the non-coding region, or it may be upstream or downstream. The interaction between these sequences and the regulatory molecules, these regulatory molecules are known as transcription factors. They induce the machinery either turn off or turn on. So what are the trans-acting molecules? They are the factors, they are the proteins. These trans-acting molecules are the proteins. Why they are called trans-acting? Because they are synthesized from the different genes, from the different DNA, from the different chromosome, and they are targeted to the cis-acting elements on the DNA. They can diffuse through the cell from their site of synthesis to their DNA binding sites. And what are the cis-acting elements? Elements mean the portion of the DNA. They are the regulatory DNA sequences. The cis-acting, why they are called cis-acting? They influence the expression of the gene on the same chromosome as a regulatory sequence. So the cis-acting elements are the DNA and trans-acting molecules are the protein. And the interaction between these sequences and molecules, such as transcription factors, can induce or repress the machinery, the transcriptional machinery. So here you are seeing that the trans-acting factors, the green colored one, they attach on the cis-acting DNA elements, and cis-acting DNA elements they will either turn off or turn off. They will either enhance or inhibit the transcription. There are the regulation, first of all, in this lecture we will study about the regulation of prokaryotic gene expression. Eukaryotic gene expression will be explained in the next lecture. In the prokaryotes, for example, in the E. coli, the regulation of gene expression, as we have already studied, that it is at the level of transcription. Transcription is the first step in the expression of gene, and the regulation at the very first step is a very excellent approach, very efficient approach, because energy is not wasted in making unneeded gene products like proteins, etc. What are operons? And we will see the gene expression in the prokaryotes by seeing the example, by studying the example of operons. What are operons? They are the genes that are coordinately controlled, that is turned on or off as a unit. This entire package is referred to as an operon. And what are the operators? Operators are the segment of DNA that regulate the activity of structural gene of the operon. At the operator, the repressor binds. As long as the repressor is bound to the operator, no proteins they are made. So if the repressor is bound at the operator, no proteins are made. If repressor is not bound, then the proteins will be made. And how the repressor will not bound? Inducer molecule, in the presence of inducer, the repressor will bound to the inducer, and so the repressor will have some changes in its structure, and it will be not available to bind on the operator. In bacterial operons, they contain an operator where the repressor binds. If the repressor binds, the RNA polymerase will not be able to transcribe the three genes, which are the LAC-Z, LAC-Y and LAC-A gene. This is an example of LAC operon system. LAC means lactose. The bacteria will use lactose as an energy source if the glucose is not present. In this diagram, we will see that what will be the behavior of LAC operon system if the glucose is present and lactose is absent. So first of all, I will explain you what are the three structural genes. These are the LAC-Z, LAC-Y and LAC-A. LAC-Z gene, it codes for the beta galactosidase, which hydrolyze lactose to galactose. LAC-Y gene codes for the permease, which facilitates the movement of lactose into the cell. And LAC-A gene codes for the thiogalactoside transacetylase, which acetylates lactose. Then in the upstream region, there is the operator region, then is the promoter region. Promoter region is the region where the RNA polymerase bind. This is the operator region, and this is the cap binding site. Cap binding site, at the cap binding site, the cap proteins which is bound, which will bound to the cyclic AMP, and it will bind. This is the cap proteins. It will bind here after binding with the cyclic AMP, but not in this case, because in this case glucose is present. For the efficient transcription, the cap binding site should be bounded with the cyclic AMP and cap. So in this case, when the glucose is present and lactose is absent, if lactose is absent, there is no need to transcribe the LAC-Z gene, LAC-Y gene and LAC-A gene. Here, in the presence of glucose, adenyl cyclase is inactive. Adenyl cyclase is inactive because glucose is present, and CAB is not bound to the cyclic AMP. So as this site will be empty, the RNA polymerase will not be able to start the transcription efficiently. Transcription is also blocked by the repressor molecule. This repressor molecule is transcribed by the LAC-I gene. LAC-I gene is a constitutive gene. Its gene product, that is the repressor protein, is always made and is active unless the inducer is present. So in this case, no mRNA and no protein they are produced. In the absence of lactose and in the presence of glucose, no mRNA and no proteins they are produced. In this diagram, when the glucose is absent and lactose is present, what happens? As the lactose is present, the genes for its metabolism, they will be transcribed. LAC-Z, LAC-Y and LAC-A, they will be transcribed. Some of the lactose molecule will change into allolactose. Allolactose, this is the allolactose. Allolactose will bound to the repressor, and repressor molecule will have will change its conformation, and this changing on conformation will prevent its binding to the operator site. Also, in the absence of glucose, the cyclic AMP will be produced as the adenyl cyclase is active, and this complex cyclic AMP plus CAP, they will bound at the CAP binding site, which is necessary for the efficient transcription of the genes. And here the RNA polymerase will transcribe its gene efficiently. Operator is not blocked and the CAP site is also occupied, and so the RNA polymerase can efficiently initiate the transcription. So this is the case where the glucose is absent and lactose is present. So now what happens if both the glucose and lactose they are present? A small amount of lactose is converted into isomer allolactose. So here the sum of the repressor molecule, it is changed, and some of the repressor molecule it will bind on the operator. And also adenyl cyclase is inactive as the glucose is present, so efficient starting of the transcription is not possible. Although the repressor may be inactive, the CAP binding site is empty, so the RNA polymerase cannot efficiently initiate the transcription. Even though the operator is off, the synthesis of few molecules of permease results in uptake and conversion of small amount of lactose to the allolactose. This is this blue color molecule, it is the allolactose, and the inactivation of some of the repressor molecule it occurs. So there is only negligible amount of transcription, and the expression is at very low level. So this was all about the LAC-operon system. Now come to another operon. This is the tryptophan operon. Tryptophan operon contains 5 genes that code for the enzyme required for the synthesis of amino acid tryptophan. As with the LAC-operon, tryptophan operon is subject to negative control. There are 2 types of its control system. One is by the tryptophan itself, which act as repressor, and another is by the way of attenuation. Attenuation is a proposed mechanism of control in some bacterial operon which results in premature termination of transcription. And this termination is by the way of attenuator, a hairpin or stem loop structure which is formed in the mRNA, and this is the same loop type structure which you studied in the row independent termination. So these are the 5 structural genes: trip E, trip D, trip C, trip B and trip A. If the tryptophan is not present, this gene is transcribed and all the enzymes which are required for the production of tryptophan, they are transcribed. But if the tryptophan is present, this will itself act as a repressor, and it will bind to the repressor molecule, and this repressor will bind at the operator region, and it will not allow the RNA polymerase to transcribe the gene. Tryptophan, it is a co-repressor because the repression by the tryptophan is not always complete. The tryptophan operon is also regulated by process, as we studied. This is by the way of attenuation. With the attenuation, transcription is initiated but terminated before the completion. If the tryptophan is plentiful and it is at high levels, the ribosomes, it quickly translates the sequence 1 and blocks the sequence 2 before the sequence 3 is transcribed. Okay, the continued transcription leads to attenuation at the terminator like attenuator structure formed by 3 and 4. Here you are seeing that there are 3 regions on the mRNA: 1, 2, 3 and 4. If the ribosome, it stalls at region 1 and 2, the region 3 and 4 will form the attenuator structure. And if the tryptophan, it only covers the region 1, then the 2 and 3 will form a paired structure, and but this paired structure is not attenuator structure. Only 3,4 attenuator will prevent the transcription. So in this case, if the tryptophan is at the high level, the attenuator structure is formed by region 3 and 4, and there will be the RNA polymerase will no longer will able to form the proteins which are required for the tryptophan. Here in this case, when the tryptophan is at the low level, the ribosome, they pause at tryptophan codon and sequences, and the formation of the paired structure between 2 and 3 prevent the attenuation. So there will no attenuation, and the RNA polymerase, it will continue doing its work, and all the proteins which are needed for the tryptophan synthesis, they will be produced. Now here we will study about the examples which say that the transcription and translation in the prokaryotes, it is coordinated, it is coupled. The first one is the stringent response. Stringent response, this is the coordination, or this is the regulation of by the way of ribosomal RNA and the protein synthesis. What is the stringent response? This is also called the stringent control. It is a stress response of bacteria in reaction to amino acid starvation, fatty acid limitation, iron limitation, heat shock and other stress condition. The binding of unchained transfer RNA means if the amino acid is not present, Then the transfer RNA is called the uncharged transfer RNA. This uncharged transfer RNA in the absence of amino acid at the A site will lead to a series production of series of events which will produce guanosine tetraphosphate. This is the guanosine tetraphosphate, and this synthesis is catalyzed by stringent factor or Rayleigh factor from the GDP using the ATP as a phosphate donor, and this production of guanosine tetraphosphate results in inhibition of RNA synthesis. However, the synthesis of mRNA for the enzyme required for amino acid biosynthesis, they are not inhibited. Guanosine tetraphosphate, it binds to the RNA polymerase and alters the promoter selection through the use of different sigma factors of the polymerase enzyme. This is the regulatory ribosomal proteins. Operons for the ribosomal proteins can be inhibited by an excess of their own protein products. What happens here? One specific ribosomal protein functions in the repression of the translation. As the process of transcription and translation, they are coupled, what happens that the proteins, they will repress the translation of the polycistronic mRNA for that operon. Ribosomal RNA protein does so by binding to the SD sequence, Shine-Dalgarno sequence, located on the mRNA just upstream of the initiating AUG codon. Some ribosomal RNA also bind to the ribosomal RNA. Ribosomal proteins, they bind to the ribosomal RNA with the higher affinity than for mRNA. But if these concentration of ribosomal RNA falls, these proteins will bind to the mRNA, so that no ribosomal proteins, they can be formed and inhibits its translation. This coordinated regulation keeps the synthesis of ribosomal proteins in balance with the transcription of ribosomal RNA. Means this coordinated regulation, there is the balance between the transcription of ribosomal RNA and mRNA. See, this is if the normal growth conditions, when the synthesis of ribosomal RNA matches that of the ribosomal proteins. Ribosomal RNA, its concentration must be in the balance with the proteins, must balance with the ribosomal RNA. This is the red DNA, it is for the ribosomal protein operon, and the blue DNA, it is for the ribosomal RNA genes. Both they are transcribed, and the ribosomal proteins, they will bound and assembled on the ribosomes. Now here, if the ribosomal RNA molecules, they are not needed or they are absent, so as the ribosomal RNA, it is absent, the ribosomal proteins, they must not be produced. So what happens, the green color one, the green colored small ball, they will bind to its own mRNA and prevent the further translation of mRNA. It tells to the mRNA that no ribosomal RNA is present, so you must need to stop here. So this was all about the regulation of prokaryotic gene expression. In next lecture, we will study about the regulation of eukaryotic gene expression. Till then, good luck. Allah Hafiz.